Hi, this will be the first of 2 lectures on protein structure. Proteins are the fourth and final of the 4 fundamental biopolymers in cells. We'll discuss that proteins are made up of amino acids. It's a linear string, like beads on a necklace. We'll talk about the peptide bond, which joins amino acids in the protein. We'll talk about the forces which govern the shape of folded proteins. And we'll talk specifically about some misconceptions about hydrogen bonding that helps determine protein structure. This slide is a review to remind you about our 2 rules of thumb that we will need to use to rationalize protein structure. The first was that opposite charges attract and like charges repel. We'll use that over and over again. This is in regard to electrostatics. And remember, this is primarily an enthalpic term of free energy. The second rule was that oil and water don't mix, and that's another simplified way of stating the hydrophobic effect, and these are mostly due to entropic contributions to free energy. This slide reminds us of the 4 biopolymers, and now we'll talk about proteins, which are made up of amino acids. Proteins are linear chains of amino acids. Individual amino acids come in 20 varieties. They are connected by bonds referred to as peptide bonds into a long linear chain. Ultimately, this long linear chain will fold up in a complex 3-dimensional shape in order that it may carry out its molecular function inside or outside the cell. Next, we'll fill in some of the molecular details of these parts. Here we can see the basic structure of an amino acid. These subunits are called amino acids because it has an amino group on one end and a carboxyl acid on the other, so amino acid. In between these 2 groups there's an alpha carbon, and connected to that is a side chain, often referred to generically as an R group. Now every amino acid is going to have this same backbone, amino group, alpha carbon, carboxylic carbon. And each type of amino acid is going to have a different side chain. Different side chains are what is going to give the amino acids their different properties. Now in aqueous solution, amino acids exist as zwitterions, meaning that they're doubly charged molecules with a positively charged amino group and a negatively charged carboxylic group. In the next couple of videos, we'll be talking about some of the consequences for its zwitterionic form. Below here we see 2 other depictions of the same molecule, a ball and chain model and a space-filling model with the amino group, the alpha carbon, and the carboxylic acid. Now I'm going to run through the 3 main different classes of amino acids based on the properties of their side chains. On this slide we have the charged amino acids. On the next 2 slides we'll see the uncharged but polar amino acids, and finally the third type, nonpolar or hydrophobic amino acids. You're going to have to remember these 3 groupings. While you won't have to remember the structures of the individual amino acids, you will have to recognize to which of the 3 groups any amino acid belongs. Here are the 5 charged amino acids. There are 3 amino acids with positively charged R groups or side chains: lysine, arginine, and histidine. Histidine's side chain can be neutral or positively charged. And there are 2 acidic or negatively charged side chains: aspartic acid and glutamic acid. All 5 of these amino acids are charged and will tend to have certain properties that you will need to be able to deduce and reason about when talking about protein structure and function. For instance, just as a preview, you would never find a charged amino acid buried inside the hydrophobic interior of a lipid membrane, but we'll get to that. The second main category of amino acids are polar, like the charged amino acids, but they themselves are not charged. There are 5 of these in total. We have asparagine and glutamine that have carboxamide side groups. And we have 3 types of amino acids that have hydroxyl groups: serine, threonine, and tyrosine. These also tend to be exposed towards the aqueous solvent. they will generally not be buried inside the hydrophobic interior of proteins. These three amino acids with their hydroxyl groups have a special chemical reactivity in that they can all be phosphorylated. This is a major way that proteins are regulated. Protein activity can frequently be turned on or off by being phosphorylated, and this post-translational modification will also play a major role in cell signaling. The third type of amino acids are the hydrophobic amino acids. There are 10 of them. They are primarily hydrocarbons, as you can see here, methyl groups, hydrocarbons, but we also have a methionine with a sulfur atom forming a thioether, and the largest amino acid is tryptophan. There are two special types of amino acids here. Proline has a side chain connected to the alpha carbon that loops around and also bonds to the amino group. Technically, this amino acid is referred to as an amino acid with an I first letter instead of A as the first letter, but that's a detail beyond what you need to know for this course. The consequence of this connection between the alpha carbon and the amino groups means that we're not going to be able to freely rotate around this bond. So that will have various structural consequences whenever proline is incorporated into a protein. And then we have the smallest amino acid here, glycine, where the side chain is merely a hydrogen atom, so that this is an extremely flexible amino acid. Both glycines and prolines can introduce kinks into the folded shape of proteins. So again, if you were on a test, you were asked, is leucine a hydrophobic or hydrophilic protein, or what class is it? You would have to know that it's a hydrophobic amino acid. This is a summary of the 20 amino acids. We're not memorizing structures, but we are again remembering the functional groups and what class they are. We recall that there are 5 charged and polar amino acids, there are 5 uncharged but polar amino acids, and there are 10, and these are the amino acids that are more likely to be involved with water or aqueous phases. Then there are the hydrophobic amino acid, these 10 here. These side chains are nonpolar, and they will tend to cluster in either the hydrophobic core of a protein or the hydrophobic section of a bilayer. Also, you are not required to remember the one-letter symbols for the amino acids, but you will be expected to recognize the 3-letter symbols for each amino acid. Now, proteins are linear chains of amino acids, and so that we're going to have to connect amino acid number 1 to amino acid number 2. And we're going to do that by a type of reaction called a condensation reaction, where we'll lose a molecule of water, but we're going to connect the carboxylic group of amino acid number 1 to the amino group of the second amino acid. So here is amino acid 1, and here is amino acid 2. They're joined between those groups in this gray box by a bond called the peptide bond. Peptide bonds are standard covalent bonds, but we refer to them as peptide bonds to speak specifically about this connection. Peptide bonds do have some properties, however, that some other single bonds don't. Even though this is a single bond, it has partial double bond character, so that these 4 atoms will be planar. You will not rotate around the peptide bond, whereas these single bonds we can rotate freely. Down here we see 3 amino acids connected. This is the amino terminus because we have the free amino end. This is the carboxy terminus. We have a histidine, a cysteine, and a valine amino acid. Here's a short peptide chain with 4 amino acids. A very important point about the synthesis of proteins, we'll see this a couple of times, is that proteins are synthesized from the amino terminus to the carboxy terminus, sometimes depicted as N to C. Here we have the amino end, and here is the carboxy end. The first amino acid in this chain is phenylalanine. Then serine was added, then glutamine, and then lysine. If we're going to add another amino acid, we're going to connect it here to the C terminus. Although the peptide bond itself is planar, we do have rotation around 2 bonds of the backbone chain. We have rotation between the amino group and the alpha carbon, and we have rotation between the alpha carbon and the carboxyl group. These angles of rotation are referred to as phi and psi, respectively. So this is, as depicted, this is an amino acid. In gray we have a peptide bond 1, in gray peptide bond 2. Remember that these 4 atoms are planar. You do not have any rotation around this single bond because it has partial double bond character. These phi and psi angles can take on characteristic values, and you can see there are certain structures that are most common. This is a topographical map showing the most common values. The red areas are the most common combinations of phi and psi angles found in folded protein structures, so that the most likely angle for phi to adopt is around minus 110. Here is 0, here is minus 180, here is around minus 110. And for psi, there's 2 structures that we'll learn about, the beta sheet and the alpha helix. If it's an alpha helix, psi will take on a value of minus 50. If it's a beta sheet, 150 will be a common value. Any of these values are possible. These are just showing the most frequent ones found in crystal structures of proteins. This is 4 different depictions of the same protein. This is a backbone-only view. It's simple to see the overall fold. Don't get fooled, however, into thinking that there might be some space in there. This is the space-filling model, where the atoms have their electronic radii filled in. And as we can see, it's quite dense and closely packed. This is basically a backbone view, except also you can see that we've added some extra depictions of regular secondary structures. These arrows are beta sheets. The helix is an alpha helix that helps our eyes draw attention to certain structural features in this protein. And here, this is in the bottom left-hand corner, we have an all-atom trace. So, 4 different ways of depicting the same protein structure. Now, with regard to forces that help govern the shape of proteins, we've got 3 really important contributors here. The first is referred to as a salt bridge, which is an electrostatic interaction. Here we see a positive charge of lysine and a negative charge of glutamic acid here. And again, opposite charges attract. So this is going to be a positive enthalpic force, contributing to the free energy stabilizing the folded protein structure. Over here we have hydrogen bonds. And as we remember, hydrogen bonds are very frequent, but a special case of electrostatics. You have a partial negative charge here and a partial positive charge here. And so it's fundamentally an electrostatic interaction. These hydrogen bonds are linear again because it's going to have repulsion from the partial negative charge here and the partial negative charge on the nitrogen. Because we're talking about partial charges rather than full charges, hydrogen bonds are not as strong energetically as salt bridges. But again, they are important contributors to the energy and final solutions of protein folding. And lastly, we have van der Waals interactions. Now, van der Waals interactions occur between any 2 atoms, but they're sort of highlighting here hydrophobic interactions of methyl groups. And we'll see how that contributes to the folding energy of a protein in this next slide. This is a schematic on the left here of an unfolded protein, with the amino acids alternating between hydrophilic and hydrophobic. And as you can see in the folded structure, all the hydrophobic amino acid side chains have gone to the interior of the protein. This burying of hydrophobic side chains in the center of the protein, away from the water, is typically the largest energetic component favoring the folded structure. Notice also that we have not buried any of the polar side chains into the interior. This slide depicts an experiment where we take a folded protein and we add a large amount of a chemical called urea to the solution. Urea is a molecule that disrupts both hydrogen bonds and hydrophobic interactions. And the result is that it will unfold proteins. That's its native structure, and here it is denatured. So it unfolds the proteins because you don't have either the hydrophobic forces or the hydrogen bonding to cause the structure. And so then we're going to remove the urea by dialysis to get the urea away. And then the protein is back in its normal buffer. And for many or most proteins, the protein will refold back into its originally native state. One very important thing that this experiment tells you is that the linear sequence of amino acids contains all the information needed to correctly fold the protein into its three-dimensional structure. There can be some complicating issues with regard to kinetics of folding and production, but for many proteins, it's simply the amino acid sequence. There are no helper proteins. There's nothing instructing it how to fold. It refolds entirely based on the amino acid sequence. The refolding process is governed by standard thermodynamics, which drives the folding of the protein. One last, somewhat minor point about this experiment. There are frequent misconceptions, both in your book and in the literature, that says proteins always fold to the global minimum of free energy. That is not entirely clear. We'll discuss this a little bit when we talk about protein folding. A folded structure is certainly a local energetic minimum and a stable structure, but we don't know with certainty if it's a global minimum. A second misconception about protein structure folding that you'll see very frequently mentioned is that hydrogen bonding is responsible for the stability of the folded protein. Let's think about that for a little bit. In a protein, you have hundreds of hydrogen bonds, and each hydrogen bond has an energetic value of between 1 and 2 kilocalories. If hydrogen bonds were responsible for the stability of the protein, that would suggest that folded proteins are potentially stable by 200 or 300 calories. 200 or 300 hydrogen bonds times 1 to 2 kilocalories. But in fact, folded proteins are typically only stable by about 10 to 20 kilocalories if you look at the energetic difference between the unfolded and folded form. And so you ask, how can that be? How can 300 kilocalories of hydrogen bonding only produce 20 kilocalories of stability? And the answer is that there are also hundreds of hydrogen bonds in the unfolded form between the protein and the water solvent. Hydrogen bonding is not a driving force for folding at all. It does help determine the final structure, though, because any final structure has to be in such a shape that every atom that can hydrogen bond does hydrogen bond. It has to satisfy all of the hydrogen bonding possibilities. If you don't satisfy all of them, you're going to favor the unfolded form of the protein. I've tried to draw that schematic here. Here's a folded protein. Every atom which can be hydrogen bonded is hydrogen bonded. So that is one correct solution to satisfying the hydrogen bonding problem. In the unfolded state, these are my poor attempts to draw molecules of water, and it's not to scale obviously, but I'm just emphasizing the fact that every single atom that can hydrogen bond is hydrogen bond to a molecule of water. If an amino group or a carboxylic acid here were not hydrogen bonded, then you would be favoring the unfolded state. So for this to be at least equal in energy, everything that is hydrogen bonded in the unfolded state has to be hydrogen bonded in the folded state. So the way I think about hydrogen bonding is that it's not a driving force for going to the folded conformation, but it is a constraint on the final possible solutions. That means only those folded shapes in which every hydrogen bond is satisfied will be likely to be in contention with the unfolded form. Again, the prime energetic driver of the folded protein structure is the burying of the hydrophobic core in the center of the protein. Now this misconception about hydrogen bonding in proteins also takes place in DNA. Here we have a C and a G base pair, and they're connected by three hydrogen bonds. A and T bases hydrogen bond with only 2 hydrogen bonds. It's known from an observed fact that DNA with high GC content is more thermally stable than DNA that has lots of AT base pairs. And then books will go on to say that DNA is more stable because 3 hydrogen bonds are stronger than 2 hydrogen bonds. But again, that's incorrect reasoning because when you have DNA that is single-stranded, all of these hydrogen bonds are going to be hydrogen bonded to water. So that's not a driving force for the folded conformation. 3 versus 2 hydrogen bonds determines the specificity of base pairing, but it is not a driving force for binding in the double helical conformation. This concludes our first video of protein structure, which we will examine more in the next video. We discussed that proteins are strings of amino acids, the peptide bond which joins the amino acids, forces which govern protein folding, and examined in detail hydrogen bonding. Thanks for listening.